And I am here to take your biology class of class 12. So, in our continuous series of classes, we are dealing with the chapter biotechnology and its application. And in current class, we are going to discuss about gene therapy and molecular diagnostics. Okay? So, when we are going to talk about gene therapy and molecular diagnostics, what does come in our mind is what do you mean by gene therapy and what is important? See, we all know that many person are born with genetic disorder. Yes, born with genetic disorder. Now, genetic disorder means there is problem itself in their gene. Okay, so which is not going to be solved by the medicine that you take the medicine and it is going to be correct. Okay, for that, because if the problem is there in the gene, then the gene that is going to code for whatever the responsible mechanisms, okay, which is responsible for whatever the mechanism that is going to be affected. As a result, the enzyme, the hormone, whatever going to be synthesized by that sequence, not going to be produced. As a result, the defect problems going to be faced by the person. So actually, scientists are moving forward to develop such method which is known as gene therapy. Means it is related, okay, it is related to the correction of non-functional gene. Okay? Gene therapy, when we are going to talk about gene therapy, actually gene therapy it is related to the correction of non-functional gene. So how this is going to be achieved? Actually what happened, near about 990, okay, in 1990, first gene therapy was performed, we can say. Gene therapy was performed in a 4-year girls. Okay, 4-year girls having ADA, that is known as adenosine deaminase. Okay, adenosine deaminase, okay, deficiency. Now, what do you mean by this? Actually, what happens, the girls is having ADA deficiency means the gene which is responsible for the secretion of this ADA enzyme, that is adenosine deaminase, is not properly formed or it's not functional. What is the importance of this ADA is that it plays a very crucial role with our immune system. Okay, it plays very crucial role with our immune system. Because so if the person is having ADA deficiency or disorder, then in this case what happens, the person may not going to survive because the immune system not going to work properly. And we all know that if our immune system is not going to work properly, then the person is going to be prone or exposed to many kind of disease. Okay, that is going to be cause problem. So what scientists do with this? Okay, as I told you, this is first trial on the gene therapy. So what happened actually? there are 2-3 methods were used, okay, but see, thing is that the genetic disorder or the mismatch, okay, in the gene is generally going to be correct. It is get corrected. I am not talking about this area, but I am talking about in the common, the gene deficiency or genetic disorder. It is get corrected either at child or embryo stage, we can say. Embryo stage. Okay. Child or embryo stage. It is most suitable at embryo stage, because in the embryo stage, actually the fetus or the baby was in the growing stage. So during that time, if the stem cells of that person can be withdrawn out and the sequence which is not functional, okay, going to be compensated or replaced by the addition of a functional sequence, okay, going to be replaced by addition of a functional sequence with the help of plasmid or vectors, okay, that is retrovectors. We have already discussed in biotechnology that in case of animal, okay, the vector we use is your retroviral vectors, okay. So that is a method, okay. Now, what are the chances taken steps taken by the scientists for the correction of this ATPase? So there are few things that can be performed. First of all, we can say administration of functional ADA, okay, time to time. Addition of or application of functional ADA, adenosine deaminase enzyme. Okay. So in this case, what happens? Periodic introduction of this enzyme or time to time introduction is necessary, because the complete curation is not possible. The complete curation is not possible. Second thing, bone marrow transplantation, we can say. Yes. Okay. Addition of functional ADA and another I told you bone marrow transplant. Bone marrow transplantation. But in both the cases, what happens? The curation is not complete. Okay, because time to time this process need to be repeat. Okay, because we all know the cells which are used is means are not going to be exist forever. Okay, they need to be time to time correction or addition of this process. One more thing here, what happens? What these scientists do? They withdraw. Okay, scientists, they withdraw the lymphocyte, okay, of the particular person, okay, because we all know though it is lymphocytes or WBC white blood cells are there which are going to be take part in our immune system. So what we can do, or what scientists did, they withdraw the lymphocyte, okay, lymphocyte of the patient. They withdraw the lymphocyte of the patient and culture that lymphocyte. Okay, culture the lymphocytes in vitro condition. In vitro condition. Okay, in vitro condition. What they do? They integrated, whatever you feel, incorporated the correct sequence. Okay, correct incorporated the correct sequence of ADA gene, okay, or ADA functional gene, we can say. ADA functional gene. They incorporated the correct sequence of ADA functional gene and incorporate into vector. Okay, incorporate into the vector, that is retroviral vectors, and transfer that to the host or retransfer to the patient. Now what happens? The vectors, what will do? They are going to be divided with the lymphocyte cells, okay, in which that they are going to be introduced. Okay, we all know here also a problem that these cells are not immortal. Yes, these cells are not immortal. So after a particular time period, this process needs to be repeated. So what actually they are trying to do in gene therapy? They want a permanent solution. Okay, but see, this is also what we have discussed till now is a great achievement. These things are also a great achievement. By by maintaining a particular gap, we can introduce any of the system. Yes, this person can survive. Okay, with this disorder. Complete curation, only there is one chance. What is that? At the embryo level. Okay, when suppose that we able to do some diagnosis, and in the embryo stage, if we come to know the genetic disorder, so at that stage, if we have got correct the disorder, I mean to say non-functional gene, by the incorporation of the functional sequence. Okay, so as at the embryo stage, the cell or the growing stage, and they have the capability to take over the functional gene, can be take over the non-functional, and in that way that can be permanently cured. But all other methods that we have discussed in gene therapy, there is a time to time incubation or treatment is required. So this is what gene therapy. Okay, so in this way scientists were working on lots of the disorders with the hope that they can be able to permanently cure the genetic disorder by recombinant DNA technology or by the deletion of the non-functional gene and insertion of the functional gene. Okay, so there are lots of research going on. So this is we have completed with the gene therapy. Okay, gene therapy. Now what next? Okay, what next that we are going to discuss is your molecular diagnosis. What is molecular diagnosis? See, in our conventional method of diagnosis of the disease, what we were doing is use our diagnosis of stools like urine, fecal matter, blood. These are the conventional method. Okay, and that also when we are able to do so all those things when symptoms appear. Okay, when symptoms appear. Suppose a person is suffering from typhoid. So when we come to know when the person is showing the symptoms of that particular disease. So it means that when a person shows the symptoms of a particular disease, it means that the causative agent already take over the body of the patient. Yes, or already they have take over the body of the host. So it is very late, and the medication, okay, curation things going to be takes lots of time. But what we can do with the help of molecular diagnosis is that suppose that you have any suspect or you have any concept that yes, you have, I think you have the chance to expose any kind of disease. Suppose that nowadays we are facing the corona problem. So in that, if you have visited any place and you get in contact with some person who are sneezing and coughing, so you may have any doubt that maybe I can get exposed. So in your doubt condition also, without observation of symptoms, okay, without observation of symptoms also, there are few methods and tests are there which can help you to diagnose if a micron percentage of the positive agent going to be present in your body. What I mean to say that in molecular diagnosis, very less or in the trace amount of positive agents are also, if going to be present in our body, we can easily going to be determine them, and at the initial stage of their infection, we can treat them or cure the body. As a result, in a very less time or in that condition, there is chances of cure of the disease, whether it is the severe case then also. So what are those molecular diagnosis methods? One, you can say PCR, okay, PCR test, okay, and another one is your known as ELISA test. Okay, so this is known as enzyme linked immunosorbent assay. Okay, and this is you all know polymerase chain reaction. Polymerase chain. Okay, now what happened here in case of PCR, as we all know that in this the fragment of gene or the sequence can be made multiple copies. Suppose that one thing is you have one paper and you make 100 copies. So what your same material you will have 100 copies. In the same way, what happens? Just pay concentration. Suppose that this is the normal sequence of the gene of the host. Okay, and if that person is infected by any causative agent, okay, so what will happen generally in the case of viral and bacteria? Okay, they are going to incorporate their genetic material also into the host. Going to be incorporate their genetic material into the host. So what happened? Scientists what did? They perform PCR tests in which they transfer or they add the nucleotides. Okay, they add the nucleotides which are radio labeled. So what happens when this process going to take place? Suppose that this is the double fragment of the DNA in between which is integrated with the infective host. Okay, infective host. So what will happen? The particular host will have the sequence of that infectious agent also. Okay, so when this is going to be separate, okay, single strand going to be doing the replication. First of all, it needs to be single strand. So when these are going to be added by radioactive isotopes, so they are going to be complementary to their template strand. Okay, those radioactive isotopes or radioactive sequence are complementary to your template strand. But what happened? They will going to be bind to the template, but where the infectious agent sequence is going to be there, they are not complementary to the template. As a result, the radioactive, okay, radio label polynucleotides not going to be bind. So on the X-ray film, okay, on the X-ray film, when they are going to be diagnosed, they will find that in a particular region there is no radioactive polynucleotides are present. As a result, they are going to do the PCR test. As a result, this is going to be produced in multiple times and they will be able to diagnose. So in this way, the cancerous cells, the AIDS patients which are infected by the HIV, they are going to be diagnosed nowadays. Okay, means this is very much important because in minute percentage of infectious agent can be determined by this method. Now another one is, I told you, enzyme linked immunosorbent assay. This test is actually antigen plus antibody. Okay, antigen antibody based test. What happens here? Suppose any foreign agent that is known as antigen, if enter in our body, our immune system going to produce special kind of proteins which are known as antibodies, and they fight back to that antigen or foreign particles. So what happens here also, if a trace amount of antigen, okay, if trace amount of antigens are going to be present, our immune system get active and the antibody process start formation. So what happens during this, a person whose blood or sample is going to be taken, which is having which is going to have the infectious agent. So in a solution, okay, a solution of gel, okay, going to be prepared, and this will have the particular antibody. This will have the particular antibody, and there is a well suppose made. Okay, in this well, the person sample was added. Okay, the person who is suffering from the disease, suppose that sample was added, and in the minute percentage, what will happen? If there is any infectious agent or antigen are going to be present, immediately there will be a reaction going to be takes place with the antibody which is going to be present in this gel or in this medium. As a result, we will see there is precipitation, there is formation of precipitation. So in this way, if micro percentage of the antigen is going to be present in the sample, the antigen antibody reactions going to be takes place. As a result, we can easily determine that yes, a person is infected with a particular kind of disease or infectious agent, and on that basis we can going to treat them. So molecular gene therapy and molecular diagnosis, in this way, with the use of biotechnology, we can cure and we can diagnose severe kind of disease. Okay, just a minute. Okay, so these are the things that we have discussed today. Okay, hope you will understood. Now the next class we are going to discuss about transgenic animals, and then ethical issues will be last topic, and after that we are going to complete this chapter. Okay, so thank you and have a